So Aliya is a second-year PhD student in the chemical engineering department. She got her master's and bachelor's degree at Johns Hopkins University before coming to LA. She has some experience working in academic lab and also in an industrial lab between her master's and coming to LA. So her current research interests lie in immunometabolism and also in studying cancer metabolism. And she's using LCMS mass spectrometry to study those. She's also involved in a lot of different organizational activities on campus and off campus. So considering all these, I would consider her to be, if I were to compare her to a metabolite, I would compare her to a ATP, which in metabolism is a high energy metabolite and makes a lot of reactions happen. So anyways, that's Aliya. And Aliya, it's all yours.
Thank you so much for the introduction, June. So my topic today is on integrating metabolomics and fluxomics to study cancer metabolism in low glucose environments. And this topic is very much in its preliminary stages, so I would appreciate any comments at the end on how to proceed with my topic after the talk. So over the last 100 years, there's been a huge growth in the field of cancer research, going from a state to where cancer used to be untreatable to now where several therapies exist is really exciting. And currently we want to continue that trend and keep on developing these new therapies to tackle different types of cancer. But unfortunately, the cancer therapy approval rate is only about 5% for therapies in preclinical development. And there are quite a few reasons behind why so very few therapies are able to succeed in clinical trials. The first is that there is still incomplete understanding of cancer biology and metabolism. The second is that these therapies have inadequate biochemical targets. And the third reason is that the models being used to study cancer biology are ineffective. The current in vitro models don't really capture the metabolic state of cancer cells, and they can't predict in vivo efficacy, especially in the tumor microenvironment. So the motivation behind my studies is to understand how cancer survives and proliferates and why therapies targeting cancer are unsuccessful to create better therapies in the future. So speaking of in vitro versus in vivo models, the tumor microenvironment is a lot different than how cells are typically cultured in a laboratory setting. Most of our understanding of cancer biology and metabolism comes from experiments in culture systems where one cell type is provided with abundant nutrients and oxygen. However, the reality is that there are many differences between the metabolism of cancer cells that are grown in culture and the tumors that are growing and proliferating in the body. The tumor microenvironment is made up of cancer cells, as well as many different types of other cells. It's hypoxic, acidic, and has poor vasculature. And because of that vasculature, tumor cells may not have access to sufficient amounts of nutrients. And so cancer cells have to have the ability to acquire and use necessary nutrients to both maintain viability and build new biomass. And to that end, they have to adapt their metabolism to survive, which makes cancer metabolism a very attractive target for new therapies. So in order to elucidate cancer survival and proliferation, we are studying cancer metabolism. So it's been known for the last 100 years that cancer cells metabolize nutrients differently than normal cells. And despite knowing this for a long time, it's been really something that's never been fully understood. So looking at the basic biology of how cancer cells metabolize their nutrients to build new cells will teach us about the new targets and new ways to go after for cancer therapy. So everyone knows that cancer cells take up a lot of glucose and convert it into lactic acid. It's the basis of PET scanning that's used in the clinic, that's used to diagnose and stage cancer. So at some level, metabolism is about microenvironments or making ATP. But if you think about what growing cells need to do, they need to do more than just make ATP. They need to make all the building blocks to make a new cell. And that aspect involves the idea of biosynthesis. Finally, the redox homeostasis counteracts the metabolic stresses that are associated with bioenergetics and biosynthesis by oxidizing NADH that's generated in the central metabolic pathways. we feel that if we can understand the difference between normal cells and cancer cells, we'd have an opportunity to make an impact on many different types of cancer. And by sort of mimicking the tumor microenvironment, we can understand how cancer cells adapt, and we could be able to target its metabolism. Our lab currently uses metabolomics and isotope tracing to study how cancer cells rewire their metabolism. Metabolomic experiments in general seek to characterize and quantify metabolites. So the first step is to measure cell growth rate and quantify nutrient uptake and secretion rates like glucose and glutamine and lactate. So these rates give us important boundary constraints on the intracellular pathway activities. But one really just can't infer metabolic activity from just changes in metabolite levels, because altered metabolic fluxes may or may not cause changes in metabolite levels. So the second step is to use stable isotope tracing to examine the intracellular fluxes. These isotope tracing studies provide information about metabolic alterations in cancer cells that really just can't be detected by metabolite levels alone. And metabolic flux studies use isotope tracers like carbon-13 or deuterium to track flow through metabolic pathways. So typically, a nutrient of interest like glucose is labeled by an isotope like carbon-13 and supplied to cancer cells in the culture medium. So a combination of uptake and secretion rates, isotope tracing, and a metabolic model can help quantify intracellular metabolic fluxes. And then metabolic flux analysis generates a quantitative map of cellular metabolism by assigning flux values to reactions in the network. So in cancer metabolomics, the stable isotope tracing and network analysis are powerful tools to uncover metabolic pathways that are differentially activated in cancer cells. So how to measure fluxes. Here we have a simple scenario on measuring fluxes. There are two contributions of carbon to form pyruvate, which are lactate and alanine. So the first carbon of lactate is labeled by carbon-13 and alanine is unlabeled. If we use LCMS to measure the labeling fraction of pyruvate, for example, we find that pyruvate is 20% unlabeled and 80% one carbon labeled. So based on that labeling fraction, we can say that about 80% comes from lactate and then 20% comes from alanine. And the ratio of the flux V1 to V2 is about 4:1. And then fluxes are also determined by fitting the measured nutrient uptake and product secretion rates to the network model. So to quantify the intracellular metabolic fluxes, we have to also quantify the crosstalk between cells and their environment. So assuming that cells are continuously dividing, the cell number will increase exponentially according to this equation, where n is the number of cells. That's typically millions of cells, and then mu is the growth rate, which is in units of 1/hour, 1/time. The growth rate is easily determined by plotting the natural logarithm of the number of cells versus time and then determining the slope of the curve. But if the cells are counted at only 2 time points, then the growth rate is determined using this equation. And finally, nutrient uptake rates and waste product secretion rates are determined by measuring changes in metabolic concentrations during the labeling experiment. So for exponentially growing cells, external rates can be calculated using this equation. And for non-proliferating cells, it's a slightly different equation using this one, where V is the culture volume and L delta C is a change in concentration. So what exactly is metabolic flux analysis? The main objective of metabolic flux analysis is to generate a quantitative map of cellular metabolism, and it's formulated as a least squares parameter estimation problem, where fluxes are the unknown model parameters and have to be estimated by sort of minimizing the difference between experimental and simulated labeling data. And this estimation is subjected to the stoichiometric constraints from mass balances and their labeling states. And there are multiple programs that sort of do this for us and computationally resolve this using something called the elementary metabolite unit framework that simulates the isotope labeling in an arbitrary network model. So going off of this example, the figure on the left shows A as the substrate for the network and F as the final product. So based on the observation of the labeling of the F molecule, we want to optimize V1 to V6. So first we input the tracer labeling pattern with the position of the labeling in the substrate. So since the second atom is labeled in substrate A, it would be 0, 1, 0. And then next we generate a random set of fluxes inside the metabolic network. So V1 equals 100 and V2 equals 110. These are all random fluxes. Third, we simulate the labeling fraction of F based on atom transition and the random flux set. And the atom transition and the flux set is defined by the elementary metabolite unit framework, which I mentioned previously. And then finally, we compare the simulated labeling fraction of F with the measured isotope labeling patterns that are obtained from our experiments. And then we calculate this error. If the error is too high, we go back to step 2 and just keep on doing a recursive optimization to eventually get solved flux values. And overall, MFA is a really powerful tool to quantify metabolism. So using these tools that we have at our disposal to study cancer metabolism, my objective is to investigate the metabolic coordination of bioenergetics, biosynthesis, and redox metabolism of cancer cells in the tumor microenvironment. We hypothesize that the tumor microenvironment plays a significant role in dictating cancer cell survival and proliferation. So to shed light on how cancer cells use the tumor microenvironment, we plan to assess their adaptability to high levels of lactate. So tumor cells, again, are highly adaptable and can adjust to a wide range of metabolic activities. So to adjust to nutrient deprivation in the tumor microenvironment, cancer cells may use gluconeogenesis. And gluconeogenesis uses non-carbohydrate precursors like pyruvate and lactate or amino acids to feed the biosynthetic pathways that branch from glycolysis. And cancer cells use an abbreviated form of gluconeogenesis by expressing an enzyme called PepCK, which links glycolysis and gluconeogenesis in the TCA. Some cancer cells have actually shown a preference of using lactate and others have not. One of the examples of that is lung tumors, which have been shown that the circulating lactate contributes to the TCA cycle intermediates. So our goal is to investigate what pathways cancer cells utilize when glucose is absent or limiting, and furthermore discern how this reliance affects their proliferation and functionality. So we started off our studies using a high lactate, no glucose environment because the TME has a high lactate concentration. Our experimental design was multifaceted. We used mouse cell lines and human cell lines that have evidence showing their capabilities of undergoing gluconeogenesis. So within the mouse cell lines, our control was the immortalized baby mouse kidney cells, and our cancer cell lines were IBMK-AKT pathway driven and IBMK-RAS pathway driven cell lines, which these two pathways are known to be really helping cancer cells proliferate and grow. And then within the human cell lines, our control was the human embryonic kidney cells, and the cancer cells were the lung cancer cell lines in the slide previous where I talked about they show some evidence of gluconeogenesis, and these two are A549 and H1299. Our objective was to model the tumor microenvironment and then conduct metabolic and isotope tracing studies to see how cancer can adapt to different carbon sources. And our first experiment was using high lactate and starving the cells of glucose. So in our human cell lines, we observed growth and proliferation in lactate medium up to a certain point. The cancer cells did not really fare well with glucose starvation. So our control cell line, HEC293T, which is the blue line here, and then one cancer cell line, which is H1299, continued growing, while A549 abruptly died off at 72 hours. For this experiment, lactate medium was changed every 24 hours. And it's interesting to note that while HEC293 took up similar levels of lactate daily, the cancer cells took up less and less each day. We want to figure out why A549 and H1299 were different. Similarly, we looked at the mouse cell lines, and we also found that they were able to grow up to a certain point, and then the cancer cells died off after some time while our control kept on growing. So some questions that arose was trying to understand why D3, which is the control line in blue, kept on surviving and what caused RAS and AKT cells to abruptly die. So to sort of further elucidate our growth rate and our nutrient uptake results, we decided to do an isotope tracer experiment using 113C lactate. So 113C lactate is an isotope tracer that can shed light on whether lactate is being used for the TCA cycle or for gluconeogenesis. So in this substrate, the first carbon of lactate is labeled. And then when this lactate substrate is metabolized, enzymatic reactions rearrange the carbon atoms, leading to a specific labeling pattern that can be measured with analytical methods like LCMS. LCMS is the preferred way to measure labeling patterns from a large number of intracellular metabolites. So after extracting our cell lysates, we run them on the LCMS to visualize the labeling patterns. So from this figure, if lactate is used for gluconeogenesis, which is going through this pathway right here, we would see labeling of N+1, so one carbon labeled in the gluconeogenic intermediates. But if lactate is used for the TCA cycle, we would expect to see little to no labeling in the TCA cycle intermediates because the first carbon would be gone via carbon dioxide. So we did see that lactate may have been used for ATP synthesis and bioenergetics because we saw that pyruvate was labeled with N+1, as you can see here, but we didn't see a lot of labeling in the TCA cycle intermediates or glutamate, which suggests that it may have been used for ATP synthesis bioenergetics. So what about biosynthesis? And we did see that cancer cells have a lot more gluconeogenic intermediates than normal cells. So we want to see if we saw some glucose and hexose phosphate in our lysolysis, and we pondered if lactate was being used to contribute to glucose production. So we also looked at labeling patterns there, but we didn't really see any glucose labeling. Like we saw literally no glucose labeling in most of our cell lines. We thought that glucose may have been coming from stored glycogen, which is broken down by the cells in low nutrient environments, and that could have been leading to some of the glucose that we saw in the previous slide. But in this side is that we also saw some serine biosynthesis by AKT and RAS in the murine cell lines. So to test our glycogen theory, we did a glycogen assay and we found that glycogen is used by all the cell lines within 24 hours of glucose starvation. And we think that the reason why cancer cells might be dying after 24 hours is that once their glycogen is depleted, their nutrient source is compromised and they are just dying off and are unable to grow and proliferate. We also looked at how metabolic flux was altered in a high lactate environment. So the figure on the left is representative of one cell line H1299 and shows a higher flux for the TCA cycle. But looking across all the cell lines, we do see that cancer cell lines in particular have a higher lactate-driven TCA cycle flux and a gluconeogenic flux. And this makes sense because they need to generate more ATP to make glucose. However, the glutamine-driven TCA and gluconeogenesis may be opposite. And another lab member is actually working on confirming that hypothesis. We also conducted some studies on oscillating lactate and glucose availability. We found that preliminary data shows that cancer cells thrive and uptake more lactate than our control IBMK-D3 cell. And we sort of want to design more experiments to study the coordination of bioenergetics and biosynthesis and redox balance. Some questions that we want to answer is where is the carbon from lactate going? And why is glucose uptake less in the cancer cells with this intermittent glucose level experiment? So similarly in the human cell lines, we saw that the control in H1299 thrived, but A549 stopped growing. We also wanted to compare H1299 and A549, the two cancer cell lines, to see what makes them differ in their adaptation capabilities. So from my initial studies, we found that cancer cells did not have a growth advantage over healthy cells in a high lactate environment, and that intermittent supply of glucose allowed cancer cells to proliferate faster than healthy cells. And we also saw that stable isotope tracing via lactate can elucidate the role of gluconeogenesis and energy metabolism. Future directions include looking at, so we looked at lactate, but questions like how D3 and Hector 9-3 cells, our control cell lines, are able to grow and proliferate are still unanswered. We could use different isotope tracers. For example, we can label glutamine in the presence of lactate to see if lactate is preferred for fueling the TCA cycle. One lab member is working on that right now, and I think they're starting to see that glutamine is more preferred over lactate. There are also many different gluconeogenic precursors like pyruvate and Aliya that can be tested to uncover preferential nutrient sources. So in addition to all that, in addition to modeling a low glucose environment, we have started our studies on oscillating glucose and lactate to study adaptation to varying levels of nutrient availability. But we still need to look at their bioenergetics and biosynthesis and redox balance behind that. All right, that was my talk. Thank you for listening. Happy to take any questions.